Protein-protein interactions are an exciting but challenging class of drug target. There are estimated to be over 300,000 protein-protein interactions, or PPIs, in humans, and many of those are implicated in disease, so they present a huge opportunity in terms of drug discovery. This is an emerging field that has been dominated by PPI inhibitors. But preventing a bad PPI from happening is only half the story. There are just as many good PPIs. Being able to stabilise those interactions with small molecule drugs would open a multitude of new potential drug targets. One example of a PPI stabiliser is rapamycin, which is a clinically approved immunosuppressant. It works by binding at the interface of two interacting proteins to stick them together, like a molecular glue. Unfortunately, designing molecules that do this is not trivial, because the interactions and interfaces involved are highly complex and dynamic. But if we can achieve that, there's the opportunity to perhaps target two proteins that don't have a well-defined ligand binding pocket. And also, the stabiliser must have complementarity with both proteins, a feature that will convey exquisite selectivity. My research group is interested in developing molecular glue design and discovery strategies so that we can better exploit them as therapeutics. We use a family of proteins called 1433 as a developmental testbed. 1433s are ideal because they interact with a range of partner proteins in subtly different ways. Stabilising their interactions is usually the logical approach. And also, we have a handful of stabilisers at our disposal that we can use as molecular tools. 1433 PPIs themselves are implicated in a whole range of human diseases. One highly relevant example is their potential involvement in the replication of SARS coronaviruses. They also play significant roles in the progression of many types of cancer. We've been actively exploring different mechanisms by which stabilising 1433 interactions might drive tumour suppression. In particular, we've been using several cell models to study the implications of stabilising the interaction between 1433 and the tumour suppressor protein p53. As part of that work, we are involved in a fragment-based drug discovery project in collaboration with AstraZeneca and the Eindhoven University of Technology. Another exciting project in the group involves developing covalent molecular glues that target a specific 1433 isoform. More broadly speaking, we've been developing a ligand discovery platform that uses combinatorial chemistry to catch and build molecular glues using the protein-protein interaction of choice as a template. We've also been developing single-molecule analytical techniques that will help guide stabiliser development. We are very keen to work with new colleagues from the pharmaceutical and biomedical sectors who can bring expertise and infrastructure in terms of computational design, screening capabilities, covalent drug development, and oncology. Collaborations such as this will be transformative in taking our research to the next phase of clinical development.